Hello, my name is Marcus Hewitt, and today is 6/21, 2001, 2021, sorry, not 2001. Right, we are going to talk about determination of gender, and the determination of gender as far as genes and hormones. All right, so we're first going to talk about the male versus female sex organs. And they basically consist of three sets of structures. And those structures are going to include gonads, internal genitalia, and external genitalia. And for gonads and males, we have the testes, and females, we have ovaries. And undifferentiated gonadal cells are called germ cells. And internal genitalia consist of the accessory glands and ducts that connect the gonads with the external environment. And the external genitalia consists of the external reproductive structures that we see. All right, we're going to talk about the human genome. And as we can see, we have 22 pairs of autosomes here, these 22 pairs right there, plus one pair of sex chromosomes, which are going to be our X chromosome and our Y chromosome. That's going to be our sex chromosomes. And sexual development is programmed in the human genome, and every nucleated cell of the body, except eggs and sperm, contain 46 chromosomes. And this set is called the diploid number. And autosome pairs direct development of the human body, form, and variable characteristics. Now let's talk about genetic sex and how the chromosomes, the X chromosome and the Y chromosome, play a role in what makes up a male or female. So, like I mentioned earlier, the two designated sex chromosomes are going to be the X chromosome or either the Y chromosome, and they both contain genes that direct development of the internal and external organs. And actually, the X chromosome includes thousands of genes that are missing from the Y chromosome. And this is basically how we see the difference between male and female characteristics with that those thousands of genes that are on the X chromosome that aren't included in the Y. And as we see here, a female who has two X chromosomes, so they're not going to have any of those Y genes that are in the Y chromosome, correct? And we see here a male parent is going to have one X chromosome and one Y chromosome. And in the sperm for the male can either contain genes from the X or genes from the Y, and that can be donated to their offspring. And the female contains two X in their eggs. Well, sorry, contains either one X in each egg, and that can be donated to their offsprings, right? So we see here if a female parent donates an egg that contains an X, and a male parent donates a sperm that contains an X, we get two X's here, which is going to make up a female offspring. And if, again, a female parent donates an X and a male parent donates a Y and a sperm, we're going to get a male offspring, the X and the Y, making up a male sex, right? And we see here, down here in the chromosome breakdown, when we have an X and a Y, we link that to a male. We have two X's, we link that to a female, right? So sex chromosomes determine the genetic sex of an individual. As mentioned previously, a genetic female has two X chromosomes, and a genetic male has one X chromosome and one Y chromosome. And on that Y chromosome, we have a sex determination that depends on the presence of the sex determining region of the Y chromosome, or the SRY gene, and that's going to come and play a huge role in the determination of sex and the characteristics we see in a male versus a female. And if this SRY gene is present, the gonads develop into testes, and if it's absent, it will develop into ovaries. So we get a female. So this is interesting because um, this is how we get the differentiation of a male and female with the presence of that SRY gene, right? That's on the Y chromosome. But in the presence of a Y chromosome that has an inactive or defective SRY gene, we're still going to develop female characteristics. So the testes will not develop and will develop ovaries and other female characteristics, and we'll talk more about that later. So here we see the sexual differentiation early development. And we're going to basically break down this whole scheme right here and how hormones and genes play a role in the determination of gender. So since reproductive structures do not begin to differentiate until the 7th week of development, sex of the embryo is hard to determine, right? And embryonic tissue is considered bipotential, so it could either develop into female or develop into male. That's what they mean by bipotential, or when we say bipotential. And that's because it cannot morphologically be identified as male or female at this point. It could go develop into female or it could develop into male at the bipotential part. And as mentioned before, the SRY gene produces protein, which is testes determining factor, or also known as TDF. And that TDF binds to DNA and activates additional genes. And that's how we get the production of the male characteristics, right? So first, let's go through this flow chart of male embryonic development here. And then we can see the SRY gene directs male development. So the presence of the SRY gene is really key, and also presence of an non-defective SRY gene is really key in the development of the male here. So we see sex determining region of the Y chromosome, also the SRY gene, produces, like we mentioned before, TDF, or testes determining SRY protein, and that initiates production of multiple proteins that cause gonad medulla to differentiate into the testes. So when this happens, we're going to develop. The testes has interstitial cells and Sertoli cells, and the interstitial cells secrete testosterone, while the Sertoli cells secrete anti-Müllerian hormone. And testosterone secreted by the interstitial cells is going to control development of Wolferian ducts into accessory structures, and also development of male external genitalia via DHT, which is a byproduct of testosterone, or alternative of testosterone, would you say. And then the Sertoli cells, which are secreting anti-Müllerian hormone, causes the regression of the Müllerian duct. And if we take this flowchart process and put it into anatomical terms or anatomical view here, we see here at this bipotential stage, or 6 weeks of fetal development, the internal reproductive organs have potential to develop male or female structures, right? We talked about that. So here we have the Müllerian duct, we have the Wolferian duct, we have our bipotential gonads here, we have the kidneys, and here we're going to break down the process, whether it's going to develop into a male or female. So let's go through the male first, since we have that flow chart over here, right? So by week 10, that SRY protein in the male embryo directs the bipotential gonads to go ahead and start developing into testes. So see, we at week 6, we are bipotential, and by week 10, the SRY gene is active, right? And it's going to begin developing into the testes. So when that happens, we start getting secretions of testosterone from interstitial cells, and also anti-Müllerian hormone from Sertoli cells. So here we see that uh, Müllerian duct starts to degenerate, and then we begin to produce, like I said, testosterone and anti-Müllerian hormone, which disappears, makes the Müllerian duct disappear. And then we begin development of the prostate, development of the seminal vesicle, vas deferens, testes, and epididymis, and we see that develop when the SRY gene is present. When it's not present, on the other hand, the Müllerian duct stays intact and actually develops further, right? So in the female embryo, absence of anti-Müllerian hormone allows Müllerian duct to become fallopian tube, uterus, and upper part of the vagina. And the Wolferian duct is the one that disappears here. So and when we have the bipotential around week 10 in men, the Wolferian duct stays and develops into the vas deferens and seminal vesicle, prostate, and all those sexual reproductive organs for the male. And the Müllerian duct is the one that degenerates. But in the female, in the absence of the SRY gene, the Müllerian duct stays around and the Wolferian duct begins to degenerate. So we get that opposite effect for the females. Okay, next we're going to talk about the sexual differentiation of the external genitalia. So in the absence of androgens, the external genitalia become feminized, right? So we see here again that we're still at the bipotential stage, that week 6, and testosterone and its metabolite DHT causes development of male external genitalia and male accessory ducts and glands, and in the absence of male hormones, the female genitalia, external genitalia become feminized. So here again, if we go back to this chart here, this kind of plays a role in the same process of the external genitalia almost at the same time. So at 6 weeks, we have bipotential, and then in the presence of testosterone and its metabolites, DHT, the male external genitalia begin to form. So the urethral folds begin to close, to form the penis. The labiosacral swellings become the scrotum. This happens at about 10 weeks, just like when the SRY gene becomes active. And then in the absence of testosterone and its metabolites, the opposite occurs. The external female genitalia begin. And like I mentioned before, that even if we have a SRY gene present, but it is defective, so for instance, we have a defective 5-alpha reductase, the enzyme that actually catalyzes the conversion of testosterone to DHT or dihydrotestosterone. So even though testosterone is present, DHT is not, because we have defective alpha reductase. And since DHT is not present, the male external genitalia and prostate gland fail to develop fully during fetal development, and this fetus appears female at birth, but during puberty, masculinization occurs of the external genitalia. You see pubic hair growth and deepening of the voice. And this is what we call pseudo-hermaphroditism. Lastly, here's my works cited for this. Thank you for listening. This has been Marcus Hewitt, and we have discussed here how genes and hormones affect determination of sex.